So thanks to both Jeff and to Jay for the introduction. I'm especially grateful for an opportunity to visit Southern California only 2 days after it snowed in Boston. So I'm very, very grateful for that opportunity. And it's nice for me to come not as the first speaker in this series, but as the third, or essentially after you've had a couple of other speakers, because what you know by now, of course, is that synthetic biology is an extraordinarily diverse field. And Jay, in talking about SYNBRC, the Synthetic Biology Engineering Research Center, said that our goal is to make biology easier to engineer. And I think what you certainly have seen with the first two talks and what you'll see today, and a departure from those, is that there are lots of different aspects of biology that we need help with facilitating the engineering. And so it's an incredibly broad and diverse field that brings in people with a variety of different foundational engineering and scientific disciplines. My background is in chemical engineering, and so that's the focus that I bring to our work in synthetic biology, with our focus being, as described here, engineering microbial chemical factories. So I'd like to start this talk sometimes. I change things around all the time. I decided to go back to a way that I used to introduce this, which is to remind everybody that from our perspective, we're not really asking biology to do anything different than it already does, which is to be a host for chemical production. And that's to acknowledge the fact that biology is actually already very good at doing chemistry. Our goal, of course, is to have that chemistry done to our specifications. But this idea of using microbes as chemical factories is actually recognizing and taking advantage of a factor that's been well known and well exploited by humans for many, many years. Now, what is different, as we've historically thought about biology for the sake of chemical production, is that we typically bring to mind very complex molecules like this. This is caspofungin, an antifungal drug with a trade name Cancytis. And lovastatin was one of the first cholesterol-lowering drugs. These are both Merck drugs, so there's my Merck bias showing on the screen there. And these are both natural products. So these were compounds that are naturally produced by organisms and which were found to have therapeutic benefits. And so from an engineering perspective, the challenge was always, how do you actually get cells to make more of these compounds? And if you look at something like caspofungin, which in addition to just having a large number of molecules, it has a high degree of functionality with ketones, with alcohols, and you can see very many chiral centers here. So we look at something like that and say, of course, you want biology to make that, given that it already does, because that's a synthesis that's going to be very challenging for chemistry. Okay? And so historically, as we've thought about fermentation, in this case, biological production of small molecules, there's been this bias towards things that are considered to be too complicated or too complex for chemistry to be able to deliver with sufficient efficiencies and yields to make it economically feasible. But I want to introduce a couple of other molecules, glutamic acid as a representative amino acid and malic acid as a representative organic acid, that are also natural products. But you can see, even though they're still chiral, they're much simpler in structure than the first two compounds that I showed. But these are also compounds that are very effectively and economically produced through fermentation, such that the supply that you currently have of glutamic acid and malic acid is not as a result of thermochemical conversions from petroleum feedstocks, but rather as a result of fermentation. So this is sort of where I came into the field. So I had a wonderful discussion with a group of students in the synthetic biology lab class earlier today. And someone asked the question, how did you get into synthetic biology? And my response was, well, it wasn't called synthetic biology when we started doing it. Okay? Our goal at this point was to try to understand how natural producers would make these kinds of compounds and to get them to do that better. And so that was called metabolic engineering. And metabolic engineering is still very actively practiced up under the umbrella of synthetic biology at times and sometimes in parallel with it. But this was really our focus. It wasn't a broader objective to make biology easier to engineer. It was, how do we get access to compounds like this that we need either for therapeutic purposes or for other purposes for our everyday lives. Again, just two other examples for historical purposes. 1,3-propane diol is shown here as a molecule noted for use on its own as an industrial chemical or as a monomer that can be polymerized into special materials, especially fibers. And this was a product that has been commercialized by DuPont. And then artemisinic acid is shown here as an anti-malarial precursor. And that was work that was done in the lab of my graduate research in Jay Kiesing's lab. And what's different about these two molecules compared to what I showed on the first slide, they're also natural products. They're also being produced today commercially through fermentation. But the difference is that the organisms that are used commercially to make both of these compounds are different from the ones where they're found in nature. So in particular, 1,3-propyldiol is made by a number of organisms, including Klebsiella pneumoniae, which is a pathogenic organism, and DuPont engineered a strain of E. coli to produce this from glucose. Artemisinic acid is a plant natural product, normally produced by something called Artemisia annua, and the Kiesling lab, working with Amyris, and you'll hear from Amyris as the last speaker of this series, worked to develop a process to produce this from E. coli first and eventually from yeast. What we do know, however, is that even if these are not being produced in their natural host, we know that the fact that they're natural products means that they are the result of a series of enzyme catalyzed reactions. And if we can identify those enzymes and then genes encoding for those enzymes, and then reconstitute those pathways into other hosts that are more genetically tractable, that is easier to engineer, then we can also have access at fermentation scales for a wider variety of molecules than we've had before. Okay, and so this is sort of where the field was when I left graduate school. I spent some time at Merck, as was mentioned in the introduction, and then came back to MIT with the idea of, or with the objective of starting my own lab. And at that time, I was actually inspired by something that I think certainly is much more on everyone's mind today than it was 10 years ago, but that's really the desire to get away from this view of the world where we use crude oil as an input and we refine that into a variety of fuels, as well as chemicals that we use for things like the plastic that covers my laptop or the fibers that are on the covers of the chairs that you're sitting on. And to think about, can we actually displace some of this crude oil with biomass as an input, or more precisely with sugars that are derived from biomass, and also then be able to produce not just fuels, but a variety of value-added biochemicals to be able to functionally replace, if not, and in some cases, a direct chemical replacement, but in many others, just a functional replacement for what we currently get from petroleum. Okay? So this was the landscape in which I started my academic career. And I wanted to think about how do we actually push biology towards making things that we don't already know that it can make. Okay, so I gave you natural products as an example where we're improving them in their natural host. I also gave you an example of finding natural products, but putting those pathways together into a different organism. We became interested in how do we actually get the cells to make things that we don't have any precedent for in nature. And we have worked on a number of molecules. I'd like to walk through today this first example, which you will see actually led to a number of other different molecules, and give you some sense about how we think about this particular problem. So 3-hydroxybutyrallactone is the molecule shown there in the upper right corner. This was first reported on the DOE's what's called the top 10 list. So the Department of Energy tried to answer this question of what would you make from biomass-derived sugars to fulfill that right-hand side of the refinery equation, where now we're making chemicals as opposed to fuels. And amongst the compounds they had on their list was HBl, or 3-hydroxybutyrallactone. And where it's currently used today is in the synthesis of pharmaceuticals, for example, in any of a number of the statin class of drugs. But the DOE report also identified this as a potential intermediate for the synthesis of new kinds of solvents, so essentially as an industrial chemical. We were interested in this because of two things. One was because the cost was fairly high. And I always have this here because it surprises me all the time what the differential can be between buying very large quantities of material. This is the best quote we could find for wholesale costs, but this is actually what we pay, about $20 to $50 per gram. We wrote a paper once where we talked about being able to use these polymeric resins to help sequester toxic fuels from a microbe. And the resins were produced by a company that I won't name. And in the paper, when we wrote it, we said, this is interesting, but we think one of the challenges to scaling it up might be the high cost of these materials. And I got an email from the company that said, oh, I just want you to know, if you bought this from us at scale, it would be about 90% cheaper because you have to pay for packaging, okay? So here's how much you pay for packaging, that differential there. The significance of this value, though, is the fact that we do know that biology can be used to make a large number of compounds. That's well established. But oftentimes, getting those compounds produced economically is very challenging. And so our original targets were not fuels, which have to have a very, very low price. But we actually tried to pick molecules where the current price was high enough that we figure if we actually could do this with biology, then maybe we would have a process that could be economical and could compete with what was currently in the marketplace. And then the last thing that made this interesting for us as academics was the fact that at the time we started this, there were no known biological routes towards either this lactone or its hydrolyzed form, which is called 3,4-dihydroxybutyric acid. Now that asterisk is there as a reminder to me. I put that asterisk there about 6 months ago because at that time, I did find that DHBA was now in a database of human metabolites, although the exact pathway for how that's produced is not known, but it's assumed that maybe it's a derivative of malic acid in human metabolism. So there is at least now precedent for this compound being present in natural biology, but at the time we started this work, it wasn't there. Now, the other advantage that you have in terms of having me here telling you this story as opposed to reading a paper about it is that you will learn as you start to write papers that when we write up science, we write the paper in the order that we wished it had happened and not the order that it actually happened. Right? We have to write it in a way that it makes sense and it makes it seem like we knew exactly what was going on and our hypothesis was exactly valid and everything flowed in place. Okay? I will tell you a little secret, which is that we spent a tremendous amount of wasted time and effort trying to figure out how do you make lactones? Aren't lactones interesting? This is a dehydration product. But yet lactones are made in biology, and how do you dehydrate in a system that's 75 to 80% water? And so we actually published a paper on making lactones in water, and that was all fine and good. But it turns out nobody really cares about this lactone. I later learned that the reason you buy the lactone as the product is because, not surprisingly, when you dehydrate this to purify it, the stable form is the lactone. If you actually want to use it for synthesis, the first thing you do is hydrolyze it. And then you actually use this as the molecule to make all these precursors that are actually useful for the statins. Okay, so that was one important thing, which was to say that in terms of chemical accessibility, the acid was just as good and in many cases better than what we would get by targeting the lactone. But what was even more important in terms of focusing on the acid was that biology had given us at least something similar to that compound in nature. And that could be a starting point for us to think about how do we actually make this molecule that we're interested in. So this compound can be thought of as a 3-hydroxyacid. This is a chiral center here. This is now a hydroxyl group at the C3 position and a carboxylic acid at the C1, hence hydroxyacids. And where do hydroxyacids come from in nature? Well, it turns out that many of these can be made as monomers in naturally occurring biopolymers known as PHAs, or polyhydroxyalkanoates. So I see some people straining. Don't worry about that. All you have to do is say, ooh, that's a lot of boxes. Don't worry about what's in the boxes. Okay? Each of those boxes, which are a lot of boxes, is a specific structure that has in common the fact that they're all hydroxyacids, but some version of these have been shown to be able to be incorporated into this diverse class of biopolymers. And biology can't naturally make all of them, although it can take precursors to many of them and convert them in the form that's acceptable to be used in these biopolymers. In nature, however, the two most common monomers that you see incorporated into these biopolymers are hydroxybutyrate and hydroxyvalerate. And while there was a lot of literature on the production of polymers that had these monomers, either as homopolymers of hydroxybutyrate or copolymers of those two, there was less that was done in terms of being able to efficiently produce the individual monomers. So we set out first to show that we could actually make large quantities of the free monomers of these. And I will again tell another truth, which is that at the time, we were doing this for something totally unrelated to the 3-HBl problem. Okay? But this being able to do this actually inspired us to figure out a solution to that. So here's now the first new trick that we want to teach our E. coli. So we're going to assemble this pathway in E. coli, and here's what our pathway looks like to get to hydroxybutyrate. We start with condensation of 2 molecules of acetyl-CoA to make acetoacetyl-CoA. And what's shown here are 3 different enzymes that are all homologs, different versions of the same enzyme called a thiolase that was known to do this exact chemistry. And what's shown in the table below now is just the sources of these different enzymes. And that's a very common approach we take, which is to say there's a certain chemistry we're interested in. We don't necessarily know up front what the best enzyme is to do that chemistry. Hopefully, we'll be able to use technology that Francis Arnold has or someone like Francis has to help us make enzymes better, but we need to have some starting point of knowing whether or not an enzyme will do the specific reaction we're interested in. So we frequently will test out a number of different candidates to try to find one out of a library that will have the activity that we're interested in. At this point, we now have to reduce this ketone to the alcohol, and we looked at 2 different enzymes to do this. The Fa-B makes only the R form, and the HBD makes only the S form. And the R form is the form that actually is the substrate for the synthase that makes the biopolymers. So you actually have to still have the CoA attached here, and then the PHA synthase will take this and make biopolymers. The S form, it turns out, is an intermediate in biobutanol synthesis. Okay, so at this point, the reason these enzymes are color-coded in red is that those are all enzymes working on exactly the substrates they were intended to work on in nature. So there was no great mystery in terms of whether or not we could actually get those precursors made to get to the final product. So the key here was to try to identify an enzyme, which turned out to be from E. coli, a thioesterase, that could efficiently cleave these CoAs from both the R and the S form and give us high concentrations of this free acid. And so we got, as you can see at the bottom, multiple grams per liter of both the R and the S form. And you can see again that for the most part, we're using a lot of enzymes doing what we already knew that they would do. If we want to extend this pathway to hydroxyvalerate, this is just a different representation of what was shown previously. The main difference here is that now we have to condense propionyl-CoA with acetyl-CoA, and then we're counting on the rest of those enzymes to go further down the pathway. Okay? So there were 2 things that were specifically different about this case versus the other. The first is that we now didn't actually have to test out several different enzymes for this first step, but instead could just rely on what was known in the literature, which was that this particular version, BKTB, was known to have very high activity for 5 carbon molecules, meaning the condensation, the assay is actually done in hydrolyzing the C5 to 1 2-carbon and 1 3-carbon molecule, but that implies a specificity as well for condensing those 2 together. Okay, so whenever we do have information available in the literature, we try to use that to help us guide our selection process to get to the final compound that we're interested in. So that was one point. The other was the fact that we have to have propionyl-CoA as a substrate. And acetyl-CoA is easy because the cells are just going to make acetyl-CoA. It's an essential intermediate. There's nothing special we have to do. Essentially, any carbon source we give it, they're going to make acetyl-CoA. Propionyl-CoA was a different matter. And you might remember that I said that this hydroxyvalerate that we're interested in has been shown to incorporate into these naturally occurring biopolymers. And in those cells that naturally make those polymers, you can simply provide propionate to the extracellular medium and you will get hydroxyvalerate monomers incorporated. Well, so we tried doing this with E. coli. We added propionate, and I am convinced the cells laughed at us. Those of you who work with cells know they laugh even though you can't hear them. And we thought, well, what's going on? So we looked at transport because there had been papers written that said that transport of short-chain fatty acids was problematic. So we got some strain variants that had regulators knocked out for transport and still nothing but a little bit of snickering. And then we finally said, well, we know that the cell does possess enzymes that will allow you to convert propionate to propionyl-CoA, but what's the motivation for the cells to do that in this case? How do we know that those enzymes are expressed? If I have glucose being provided here to give me nice glycolytic flux to acetyl-CoA, there's no reason for the cells to have to metabolize propionate. So we decided the problem was not propionate uptake in the cell, but rather having sufficient flux to propionate-CoA. So instead of relying on endogenous activities for this, we actually identified other enzymes from nature that we knew could take propionate and turn it into propionate-CoA. And we looked at actually 3 different systems, but I have 2 that are shown here. One which is from the same organism, Clostridium acetobutylicum, that makes butanol. And this pair, the PTB-BUK system, is actually implicated in butyrate metabolism in that organism. And it will give you propionate-CoA from propionate through a phosphorylated intermediate. The other enzyme is called PCT, which stands for propionate-CoA transferase. And in this case, it will take a CoA from acetyl-CoA such that you start with propionate and acetyl-CoA. You end up with propionate-CoA and acetate. Okay, so our main hypothesis here now is that as long as we had high activity or high expression of these enzymes, we would have sufficient propionate-CoA in order to get the rest of the pathway to work. So here's what the data look like. What I have shown here now are the red bars are hydroxybutyrate. So you might recall we're using the same enzymes that we use for making hydroxybutyrate. So we've got a competition now between 2 acetyl-CoAs binding and acetyl-CoA plus propionate-CoA binding together. And without any of our activation enzymes to make propionate-CoA, you can see large amounts of hydroxybutyrate. Then these dark blue bars are the hydroxyvalerate. So there's very little of the hydroxyvalerate being formed. And I have the molar ratios of the C5 to C4 plotted or calculated below, just so you can see it really is a small amount of material that's being made. If, however, we have either of our 2 activation mechanisms, and I won't go into the details here, the important thing to see is that in all cases, the blue bars go up significantly, the red bars come down. And in our best case, we actually get a 12-to-1 ratio of preferencing the C5 over the C4. You'll see this also comes with a tremendous amount of acetate, and that's because this PCT that we use is the one that gives us acetate as a byproduct. And so if that system is actually working really well, it turns out we have a lot of acetate, and we're still working on how to deal with that excess carbon flow. So the take-home message here was that you have to activate these short-chain acids, propionate in this case, and exactly how you activate them is going to determine the product distribution, both in terms of absolute titers, but more importantly in terms of the ratios or the selectivity that you get between the desired C5 and the C4. Okay, so I said we were doing this for totally different reasons, and I'm not going to have time to go into why we were doing that. But at the time that we actually were successfully making these hydroxyacids and were fooling around with a bunch of lactones that didn't really matter, we realized that here is chemistry we had already demonstrated where we could condense acetyl-CoA either with itself or with a propionate-CoA molecule, and we could get all the way to the free acid. And if we looked back at the structures we were really interested in, it wasn't really that different. So here's 3,4-dihydroxybutyric acid. And if this same pathway is going to work, all I need is for one of these enzymes to be able to condense acetyl-CoA plus glycolyl-CoA, that's this molecule, to give me now this hydroxylated beta-ketovaleril-CoA. And then I need one or more of these 2 reductases to still work on reducing this alcohol, this ketone to the alcohol. And then I need my thioesterase to be able to cleave the CoA. Right? So now I'm starting to ask the enzymes to work on molecules they haven't seen before, but they're not that different. And so we have some hope that maybe we can actually get the right activity that we need to see the product that we want. Then there's a matter of this little molecule here, which again I said is called glycolyl-CoA, which is essentially glycolate plus the CoA added to it. And I didn't really know where to get glycolyl-CoA from, but I knew we could buy glycolate from Sigma. And we showed with the propionate case that we had enzymes that were capable of adding CoAs to propionate. So maybe that same enzyme would have enough specificity or flexibility in its substrate range to be able to activate the glycolate as well. So here's what the result of that experiment looked like, where we now have glucose and we have glycolate in the medium as well. We're testing now in that first step all 3 of those enzymes that we know can condense 2 molecules together again, because now we're looking at a system where we don't have any evidence that it will work specifically on these 2 substrates. So we're opening up the toolbox, if you will, to try to explore a broader range of enzymes. The three that are grouped here are for one of our two reductases that gives us one of those two enantiomers in terms of that chiral center that's set. And then this set of three is the other reductase. The PCT is the enzyme we're using to put the CoAs on to glycolate. And then the TESB is what's used to take the CoAs off of the 3,4-dihydroxybutyryl-CoA. And what you can see, again, as I've been talking, is that we actually managed to make more than 0.5 gram per liter of the free acid in this case. We also got about 200 milligrams per liter of the lactone, and we actually know where that's coming from, which is that it actually comes from the version of that molecule that still has the CoA attached. And because the CoA is a good leaving group, that C4-hydroxyl can give you a nucleophilic attack and close the ring. What we don't know is if that's something that's happening spontaneously or if it's catalyzed by a specific enzyme in the cell. But for our purposes, we don't really care because we know as a result of the purification, all of our acid is going to go to the lactone anyway. Now, I'll point out here that the stereochemistry is a little bit different. This is now the S form rather than the R form because the presence of that hydroxyl group actually does change the ordering of the atoms. And so now we went from being R-specific to S-specific. And the nice part about this is that it's actually this form of the molecule which is useful for statin synthesis. So we sort of had a 50-50 chance in terms of getting one of these to work well, and it just so happened to be the one which is useful for one of our intended purposes. So we proved out that we had this. The reference was shown on the previous slide, which was that this was published in 2013. We first made this molecule about 1.5 years before then. And it took us a long time to publish it because something was wrong. And this is actually one of the nice advantages of having a lot of turnover in a lab because people come and they graduate and they leave. And by the time you've been in your 4th or 5th year, you're really jaded and you're like just ready to go and you're not really paying close attention anymore. And a new student came in the lab and said, I think that there's another molecule there. And it has the same molecular weight as the compound that we are trying to make. It's only produced when glycolate is present. It's only produced when all of these enzymes are present, but it's not the same molecule. And he was pretty sure about this because there was a slight misshapen in the HPLC trace, and then I'm wondering, did he have too much coffee or too little coffee or too much beer or too little beer? And he was, however, able to get 2 distinct peaks on a mass spec. Okay? So we sat around and we did a little bit of brainstorming trying to figure out what could this be. And then we went to the mechanism, which is a hard thing for me to do because I'm an engineer, not a biochemist. Right? But we went and said, okay, how does this enzyme actually work? And for the molecule that we're trying to make, which is shown here, for this first enzyme, what happens is that you bind glycolyl-CoA, and then you actually will abstract a proton from acetyl-CoA, and the resulting carbocation catalyzes a nucleophilic attack on this carbonyl group here, and that's how you actually get now that carbon-carbon bond being formed. Okay, so then we ask, well, again, what could have the exact same molecular weight? It depends on all these same things being present, and we decided that what probably happened is that the order of this condensation reaction got flipped. So that rather than having glycolyl-CoA bind and then have the acetyl-CoA proton abstracted, you actually had acetyl-CoA bind first, which we know is possible since 2 acetyl-CoAs can come together. And now we're actually getting a proton abstracted from glycolyl-CoA. And if I have nucleophilic attack here, what I get is an isomer, which results in 2,3-dihydroxybutyric acid instead of 3,4-DHPA. It would have the exact same molecular weight. It would depend on having those same enzymes as well as the same precursor. So the way we decided to investigate this was to use labeled substrate. And this was another case where I thought I knew what I was talking about, but I didn't, because I said, well, let's get C13 labeled glycolic acid, because then we can at least prove that whatever is being made actually has glycolate in it. And the student came back and said, I can't get that, but I can get deuterated glycolic acid. And it's fortunate that that's what he could get, because if you look now at the mechanism, if the first compound is being produced, we'll hang on to both of these deuteriums, and we'll get a molecular weight of 140. If instead the second compound is being produced, we'll actually lose one of these deuteriums, and the molecular weight will be 139. And that's exactly what happened. And so it turns out we're making both of these. And at first, we were sort of disappointed. And as the first student who had graduated by now sent an email saying to the second student, you have now officially disproven 50% of my thesis. Because roughly half of what we make is actually the other compound. And we already knew that there was a point or a purpose for this first one. So then the question was, we've got a lot of this other one being made. Is this just wasted material, or is there some utility that can come from that? And that's what makes it nice to be in a place where people talk to each other and can collaborate very easily. And I have a colleague whose name was actually on the first slide that had that biorefinery picture, Brad Olson, who's a material scientist. And Brad said, I think this is a very interesting molecule for new kinds of materials, because that carboxylic acid gives us a place to do polymerization, but you now have these hydroxyl groups that give you handles for doing other kinds of chemistry. So we now actually have two projects coming for this. One where we're trying to improve the synthesis of this compound. The other where we're trying to selectively improve the synthesis of the other monomer so that we actually have two different end goals in terms of how we're using these. So here's what our pathway looks like overall. This is the numbers that I showed you before in terms of how much of these compounds we were making. We do still make hydroxybutyrate. And it's a byproduct that we're always sort of faced with because of the fact that those enzymes can still condense two molecules of acetyl-CoA. Although I will say that we've tested a number of different enzymes since then, and we do see some differences in the selectivity between those two. And I alluded to this previously, which is that this 2,3-DHBA, the amount that we make is roughly equivalent to the combined amount of the 3,4-DHBA and the 3-HBl. So that enzyme that's doing that initial condensation reaction has very little selectivity between those two different forms of the enantiomer. And the last bit of this story I wanted to tell is the fact that instead of feeding in glycolate, we actually know that there's a pathway where we can make glycolate from glyoxylate in the glyoxylate shunt of metabolism that's present in E. coli. And so we recently published this paper showing how we could actually get all of these compounds from glucose as the sole carbon source. So I do have a second story to tell. It's much shorter than the first, so don't get concerned. I'm sleepy myself because I'm 3 hours ahead of you guys, so I'm not going to stay here all night, trust me. Here's where we started. We can get a couple grams per liter of 3-HB. We can also get a couple grams per liter of 3-HV. But we were interested in whether or not we could extend this. So if we fed in longer chain acids or branched acids, could we actually get now other kinds of structures? And so if we fed in butyric acid, we could get the 6-carbon hydroxyhexanoic acid. But two things happened. One was that you see we make a lot less of this compound, only 40 mg per liter compared to about 2 or 3 grams per liter. And we're only getting one of the two enantiomers being produced. Okay, so we don't know at this point if the problem here is that we have the HBD being non-functional, or maybe we're now starting to get into some selectivity issues with the thioesterase preferentially hydrolyzing one versus the other. If instead we used isobutyric acid to get hydroxymethylvalerate, then we did a little bit better. We still only make one of the two enantiomers here, but now we're up to about 300 mg per liter. So we're only a tenth as opposed to 1/50th of what we had with the other ones. And I have this in blue here because I'm going to tell you a short story now about taking that and trying to address some of the biofuel side of the refinery. So our interest, again, has been in trying to push biology to do things that it doesn't normally do. And so when we work on biofuels, we don't like ethanol. Ethanol, we know how to make ethanol. We want to figure out how to make things that we don't know how to make. And so we actually had a project that was sponsored by Shell where they had a specific interest in getting to branched molecules because the energy density here is a lot higher and the octane numbers are a lot higher as well. So you can see compared to ethanol and butanol, you get about 97% of the energy density of gasoline with 4-methyl pentanol. And you also have, I don't have the numbers here, but the octane rating is very, very high. So this actually looks like something that could be a useful fuel. And if we just do some calculations about energy efficiency as well, if we mimic the pathway that exists for N-butanol biosynthesis, it suggests that if we actually can get the titers up, we can do this in a very efficient way as well. So this now is a much more complicated pathway, but I want to remind you that we had enzymes that allowed us to put CoAs onto things and take CoAs off of things. And that actually allows us to take a complicated pathway like this and split it up into different pieces. So the core of this is the first part, which I showed you already, which is that we can condense isobutyryl-CoA with acetyl-CoA, and then we actually eventually completely dehydrate that to get down to 4-methyl valerate. And this is just a reminder of the fact that we always have this competing reaction of the two acetyl-CoAs coming together. Once we have now the free acid, we have to be able to reduce that first to an aldehyde and then down to the alcohol. And I'll go over the details of how we did that in just a second. And we ultimately want to be able to do all of this from isobutyrate, so that gives us isobutyryl-CoA, but eventually just from glycolysis or some carbon source that allows us to make isobutyrate from another precursor. So we want everything to be able to come from glucose or glycerol as a sole carbon source. So the first part of this story I already told you in that summary of extending the hydroxyacid pathway, which is that we could actually shortcut this pathway using this test B enzyme from E. coli to get to 3-hydroxy-4-methyl valerate. And what I said on the previous slide was that we got titers of about 300 milligrams per liter if we use this FAB gene. Here's just the data showing that we actually don't get anything if we're using the other reductase. But what I didn't show you on the previous slide is that we make 4 times as much hydroxybutyrate as we do this hydroxy-4-methyl valerate. And so we're making the compound that we want, but we've got a selectivity problem. So our challenge here was both making sure that we could have this precursor be reduced all the way down to the alcohol, but also trying to do that by selecting enzymes that would be gatekeeper enzymes, ones that would selectively pass through those molecules that get us towards the final product that we're interested in. So how do we do that? Well, the next step after we have this hydroxylated compound is to actually dehydrate that and then saturate it. And at this point, we actually didn't have enzymes that we knew could do this chemistry. The enzymes that work for butanol biosynthesis will only work if you use that enzyme that gives you the S form, and that was an enzyme we knew didn't work. So instead, we went back to the literature again and found enzymes that were implicated in biopolymer synthesis once again that could do this kind of chemistry, and then another set of enzymes to do the second step, so that we had a total of 24 combinations. Again, we didn't know which ones would work and which ones wouldn't, and so all 24 combinations were tested, and of those 24, 6 were able to give us 4-methyl valerate as the final product. And you can see as well that we get roughly equivalent amounts of butyrate in all of these systems too. Okay, so we've already achieved our first objective, which is that we're getting some selectivity of the pathway. When we started, we were 4-to-1 in favor of now the 4-carbon molecule. Now we're roughly 1-to-1 with equal balance of the C6 and the C4 molecule. Okay, and we believe that this is really being controlled by the selectivity of these enzymes. So the next step of this then is to get from this 4-methyl valerate that was shown on the previous slide down to the alcohol. And in natural N-butanol biosynthesis, what happens is that you don't actually cut those CoAs off. I alluded to that previously, but instead, with the CoAs still on, the molecule is reduced to the aldehyde and then down to the alcohol. Well, we were able to test these enzymes in vitro, and we were able to show that they wouldn't work for these particular substrates. So we actually had to find another set of enzymes. So we decided to look at an enzyme called carboxylic acid reductase, or CAR, to do this first step of converting the acid to the aldehyde. And then the last step, in terms of converting the aldehyde to the alcohol, we started with a very broad activity alcohol dehydrogenase called 88-6 from yeast, which has really been shown to work on pretty much anything that anybody has ever tested it against. It works on many, many different molecules. So this CAR enzyme, there was some literature on a different version of the enzyme that had been tested and the selectivity had been profiled. The particular version that we had not been tested, and so we purified that and looked for activity against the substrates that we were interested in, as well as other substrates that were of similar size. And we found something very nice, which is that here's our target compound, 4-methyl valerate, and you can see that that's actually where we see the maximum activity. The two major competing substrates we think should be present in this system are butyrate, that comes from condensing the 2 acetyl-CoAs, and isobutyrate, which we're either supplying or which we're producing from glucose. And so the activity here is actually much higher on the compound that we want than on those competing compounds. And if we actually calculate Michaelis-Menten parameters, you can see that there's a much greater specificity or preference for this longer chain 4-methyl valerate than there is for the isobutyrate. So that looked very promising as well. And so if we put all of these together, where we feed in the isobutyrate, we have a way of activating this, and then ask how much alcohol do we get at the end, you can see we get a decent amount, about 300 milligrams per liter of 4-methylpentanol. We also get now a significant amount of butanol, but we're shifting selectivity even better in our favor. So we went from 4-to-1 against to 1-to-1 to now 2-to-1 in favor in terms of 4-methylpentanol and butanol. You can still see some isobutanol being produced, and we actually see butyrate building up, which is again an indication that this CAR enzyme is selectively converting the 4-methyl valerate over the butyrate. So the last part of this was to get a way for us to make isobutyrate from glucose, and there had been a pathway that had been well documented in terms of being able to do this kind of chemistry. And the key in this case now is that this pathway at this step actually involves the oxidation of isobutyraldehyde to isobutyrate. And that's great, except that we want to make sure that we don't have the competing reaction of now the 4-methyl valeraldehyde that will be generated here also being oxidized back to the 4-methyl valerate, because that would just create a futile cycle for us. So we would oxidize and reduce and oxidize and reduce. So in the literature, there had actually been 4 different genes that had been tested that were supposed to be able to produce now this isobutyrate from isobutyraldehyde as an intermediate. And when we tested all 4 of those, only one of them, this gene that's called FIA-B, was able to produce 4-methylpentanol. And you can see here we get about 180 milligrams per liter. We still have now some butanol, but you can see the selectivity now has gone way in favor of the butanol. We do, however, still have some 4-methyl valerate that's being produced, and so that led us to think that we were actually having this side reaction of the 4-methyl valeraldehyde going back to that acid. So instead, again, going back to the literature, we identified an enzyme from another bacterial system which should have much greater specificity for the isobutyrate and a lot less activity on the 4-methyl valerate. And you can see what happens in this case is that we have no residual 4-methyl valerate, and we again get even less butanol being produced. So this is all great in terms of the selectivity. The only problem is that we're still making a lot of isobutanol. Okay? So we had great selectivity for 4-methylpentanol over butanol, but you can see here that we have a significant amount of isobutanol being produced, about 4 times more of that than we have of the 4-methylpentanol. And those are just the same 2 cases shown on the previous slide. So if we ask where that's coming from, then we have this ADH6, which is very broad activity, which we want to be able to produce the 4-methylpentanol, but which we think is also just taking the isobutyraldehyde and converting that to isobutanol. And so to address this, we can first look at what our titers are with the core system that we had before. And you can see now, again, 4-methylpentanol. Here's now the same data here, so you can see it on the same scale where we have significantly more isobutanol. And the last step here then was to identify a specific alcohol dehydrogenase now from a different species that has much more specificity for the longer chain substrates over the shorter chain ones. And you can see that the titers of isobutanol go way, way down. The titers of our target compound go down as well, but we went from having a selectivity that was 4-to-1 against what we're interested in to having a system where 80% of all the alcohols in the system, that's 4-methylpentanol, that's butanol, that's isobutanol, 80% of everything we measure is actually the compound that we're interested in. So let me summarize. We started with this pathway for the production of a number of 3-hydroxyacids, and that actually led us to identify these systems to put CoAs onto molecules and to take CoAs off of molecules, which really allows us to have the flexibility to test pathways broken down into individual pieces. We were able to mimic the pathway that organisms naturally use, the so-called ABE pathway for N-butanol, to get this new compound, this C6 branched 4-methylpentanol. And I went over a lot of different genes here, so I just want to summarize here that the final pathway actually takes genes, it's 10 different genes from 8 different organisms, none of which are actually designed to make these alcohols at all. It really is just relying on what we know about chemistry in the ability of these enzymes to do chemistry to get to the products that we want. And the challenge that we have now, of course, is that this is proof of concept. So we've shown we can make some molecules, but we don't have nearly high enough productivities or selectivities to get us the titers and yields that we need to really be feasible. And this is where I always say now the fun really begins. And fun is in quotes because, my job now is to go around and talk about the work that other people do. So, it's fun for me, but I don't have to work in the lab anymore. So I don't know how fun it's going to be for the students. We'll see. It depends on how successful we are in the end. So I just want to end by acknowledging the folks who did the work. They're color-coded, so the former students in blue and Kat, a current student, were all those students who work on the original production of that dihydroxybutyric acid and some of those homologs. And then the biofuels work that emerged from that was work that was done by a former postdoc and graduate students. It's being continued by a current graduate student, Ditya Kunjapur, in the lab. The funding sources for this work are highlighted in green here. And then I have this just as a standard slide, a standard statement on all of my acknowledgement slides now. In this particular case, that first pathway that I talked about for DHBA has actually been licensed to a startup company. So I have to declare competing financial interests. And so with that, I am delighted to take any questions, and I thank you very much for listening.